Okay, so our next speaker is Dr. Rohan Paul. Dr. Paul joined us from George Washington University. He joined us last year. He had a few years of experience there. He came with experience and expertise in diagnostic tools, including cell-free DNA and molecular diagnostic. And he is now working on single cell analysis and trying to expand further in this regard. Today he will talk about molecular diagnostic in kidney transplantation.
Well, good morning. Microphone's working. Thanks, Dr. Ahmad, for the kind introduction and for the invitation. So I don't have any disclosures for this particular talk. My only disclosure is that to be factual, I'm not a molecular biologist by training, so everything I'm presenting is through a clinical lens. There we go. So I'll just put this slide up more or less to set the stage for this particular topic. I think we're all familiar with some of the challenges in achieving desirable allograft outcomes, whether it be in the form of diseases that compromise the longevity of the graft, infections, rejection, so on and so forth. I think everyone is familiar with the limitations in current surveillance strategies for allograft monitoring in the form of creatinine or DSA. And I think we all appreciate the challenges associated with getting a biopsy, and sometimes not that unoften, the limitations in actually interpreting a biopsy. And then I think the other thing is there are knowledge gaps or a lack of tools to adequately guide us to help us gauge alloimmune risk at a particular point in time, in real time. And that obviously leads to the risk of inadvertently over-immunosuppressing or under-immunosuppressing patients, because we can't gauge what's an adequate amount of immunosuppression for a particular patient. And with that comes the risk of infections, rejection, or other complications that also work against us. So that really sets the need for real-time biomarkers and the need for high-precision molecular diagnostic tools, even on top of the tools that we already have. So the biomarker classification system and the process for biomarker qualification is complex. But features of an ideal biomarker, if we were going to reflect on it, would be one that's safe and easy to measure, one that has a rapid turnaround time that enables us to make a diagnosis and instigate a plan of action, one that's not cost prohibitive, one that's accurate, consistent, and ideally one that's also specific for a particular disease. And this is where it becomes challenging in allograft management or transplant medicine, because these diseases are constantly being defined and redefined as the BANF classification system evolves, and it should as we learn more. And as of now, one of the key areas that molecular diagnostics is being targeted towards is the detection of alloimmune sensitization and activity. And what I'm really getting at is that most of the or many of the diagnostic panels that are being developed are really gauged towards detecting the T-cell activation event upon antigen presentation. This is a busy diagram from a nice review by Saffer and colleagues. This is about 6 years old now, but it's still quite accurate and quite well-encompassing. Basically, it just summarizes the key intracellular and extracellular events that take place upon T-cell activation. And it also alludes to some of the diagnostic tests that have been developed to detect it. So, and I know it's busy, but some of these are already standard of care, such as the ones in red, like the cross-match or Luminex for DSA. And it also alludes to some of the gene expression assays, either the targeted single genes or the gene panels, which are shown in the green box, the array-based. And on top of this, something not mentioned in this diagram, which I can add, is we can also add molecular diagnostics of allograft stress in the form of cell-free DNA, which I know you're all familiar with. And we can also add some of the newer technologies like nanostring and RNA-seq. So as far as sequencing platforms in history goes, there's been significant evolution since the development of the Sanger technique nearly 50 years ago. But the real point from this slide is that it's really been with the availability of massive parallel high throughput sequencing that we've been able to leverage this technology in clinical medicine, including transplant, because we can achieve an adequate turnaround time that we can make a diagnosis and develop a plan of action. So the crux of RNA sequencing, just the very basics, it's really the ability to generate circular DNA libraries which we can then amplify and then sequence with next-gen sequencing and then map to a region of interest on the chromosome. And with the emergence of those tools has come the emergence of various, with the emergence of those technologies has come the emergence of various tools. And in the interest of time, I'm just going to be limiting myself to these three. So cell-free DNA, historically quite interesting, nearly described nearly 70 years ago by Mandel and Mateus, and it's histone-bound with a short half-life. And its potential in solid organ transplant was first described by Lowe and colleagues in 1998. And that vision has now become realized through the availability of high-throughput sequencing, which is what I was trying to get at. And there are various detection strategies that have been trialed to delineate DNA from person A to person B. But the one that's commonly used for most commercially available assays is genomic transplant dynamic methods. And what GTD methods really is it exploits the fact that solid organ transplant is effectively a genomic transplant. And within and between those genomes lie single nucleotide polymorphisms in key regions of the genome that can help us distinguish one organism from another. That's still prone to computational error because in a living related transplant there may be similarities at key loci. So in 2017, Sharon and colleagues actually proposed a mathematical model that eliminates that dependence on the two genome approach and has made this computationally more accurate. And in terms of the assay itself physically, it's really built upon a curated panel of primers that are targeted towards these SNPs of interest, and those primers are different for the different commercially available assays. Following DNA amplification, there's next-generation sequencing via something like the Illumina MiSeq platform and then the generation of raw data. And that raw data is then subjected to a customized bioinformatic pipeline, which is also different from the different protocols and assays. But the principles are generally the same. There's alignment, and with a reference genome, there's filtering of ambiguous reads, and then there's extensive quality control steps that ultimately culminate in the generation of an allelic count. And that allelic count can be spitted out as either as a absolute number or as a percentage. So of the some of the big ones, AlloSure, Prospera, Viracor, we've used AlloSure and Prospera at our centre, and I used those at my previous centre as well. And this is just a table from a review we published several years ago, comparing AlloSure and Prospera. It just points out the differences in the number of SNPs. And Melanson and colleagues from GW and DC reported a slightly higher sensitivity with Prospera and a slightly higher specificity with AlloSure, but found them to be fairly comparable in his group's analysis. And that was corroborated by Roy Bloom and Tara Sigdell at UCSF. And then in terms of clinical utility, this is a growing area, and there's a multitude of papers that are still being published. But the very basics, there's a wide range of variability immediately post-surgery, partly because of ischemic reperfusion injury and partly because of limited glomerular filtration post-transplant. There is some data that cell-free DNA may even predict the duration of, effectively the duration of DGF post-surgery. Bromberg's group in University of Maryland found that at a cutoff of 1% for cell-free DNA, that was associated with the 96th percentile of all results. And then we're all familiar with the DART study by Roy Bloom at Penn using AlloSure, and found that the highest percentage of cell-free DNA was in the AMR category. And then using the other panel, which is 13,000 odd SNPs, which is used by Natera, that was associated with, that had the highest percentage in and among those with acute cellular injury. but still higher than those with borderline rejection or other forms of injury or stable allograft function. And then just recently, in fact, Dr. Al-Hamad and his collaborates found that at a cutoff of 0.5%, and with a delta increase in cell-free DNA, that there was an association with rejection, and it may also predict the onset of de novo DSA. One study reported that there might be superior value in using an absolute number rather than percentage, although there's been variation on that. And then the trifecta, the information from the trifecta study is probably easy just to show on the bar chart. This was interesting because it looked at allogeneic injury, biopsy proven, both histologically and in terms of molecular markers, which I'll get to in a moment, and looked at the degree of cell-free DNA that was associated with it, and found that histologic markers of AMR was most associated with cell-free DNA elevation, as you can see in the blue bar, so PTC lesions or G lesions. And then the same applied, the same diagnostic categories seemed to rear their heads when looked at it molecularly, again shown in the blue bars. And then just moving on to microarray, I guess the principles of microarray is that it's gene expression detection, first with mRNA isolation, followed by circular DNA library generation, which is fluorescently labeled and then incubated on a DNA chip or a grid, which contains complementary DNA probes corresponding to the various genes of interest. And that circular DNA is then visualized, quantified, and analyzed. And historically, one of the earlier assays was developed by Mini Sawal at UCSF, which is the K-sort assay. This utilized peripheral blood as its tissue of interest, and looked at 17 genes that were involved in immune cell activity. And it was able to, in their study in 2014, it was able to predict rejection with adequate sensitivity and specificity, but couldn't discern the rejection type. Another group reported that if combined with ELISpot, which is an interferon gamma release assay, that diagnostic accuracy could potentially be increased. However, in a completely independent undertaking by Martin Naessens in Belgium, along with his European collaborates, the K-sort didn't have diagnostic value for detecting acute rejection. And then the other big hit on the block, microarray-wise at least, is the molecular microscope by Phil Halloran in Edmonton and produced by One Lambda. In contrast to K-sort, this actually utilizes renal allograft tissue. And as you can see, it's a lot more probes, a lot more genes, something like 19,000 odd. And it utilizes ensembles of predefined machine learning algorithms and compares that to a reference set. And it's based on, it utilizes pathogenesis-based transcript sets, and it's based on the premise that pathologic conditions are reflected in gene sets rather than in individual genes. And just on that table on the right, you can just see a snippet of some of the PBT sets that are used. And then this is just a diagram taken from their review on MMDX heart, but it's a similar workflow for kidney, which starts from the collection of the tissue to the processing, and then ultimately to the generation of the MMDX report. Things to be said about this is that biopsies need to be stabilized in a special solution because paraffin fixation may damage the RNA. And as I alluded to before, the biopsies are compared against a reference set using a predefined algorithm. And disease states are regarded as latent variables or unknowable truths. And then phenotype labels are assigned by clinical experience and histology. And while molecular markers are deemed to have the highest weight in defining that category, some algorithms are trained with correlation with histologic data. I'm going to have to move a little bit quickly. There's numerous studies that define the clinical end, that have used MMDX in clinical endpoints. This is an interesting study that's actually going to compare MMDX with other parameters that's ongoing in Rush. And then I think in the interest of time, I'll skip over TrueGraph. Yeah. I'll just touch on NanoString. This is the other assay, and without going into the technical details, in a collaboration with the Banff Molecular Diagnostics group, they collaborated with NanoString. So this is commercially available but non-proprietary, and utilizes 700 odd genes as opposed to 19,000. And these were genes derived from peer review. This is just a table that I'll probably finish up on because it's important, that comparing NanoString with microarrays. As you can see, a very big difference in the number of genes that are involved. But practically important things to be said is that it does depend on fresh tissue with microarray, whereas with NanoString it can be done on paraffin block tissue. And it's also less steps because there's no reverse transcription involved, and it's cheaper, much cheaper. And these are some studies that are attesting to the potential utility and the correlation with microarrays. And then I'll leave this slide up because it summarizes what I've already said, and I'm happy to take questions.
Thank you.
Thanks.
Thank you. It's a tough, broad topic, so thanks for summarizing that in 15 minutes. We'll take one question. Any questions? Yes.
Thanks, Dr. Paul, for your talk. I just wanted you to comment on the recent Medicare, CMS ruling. Because interesting comment, they said that these tests are costly, but there was no comment about value. So just wondering if you have any thoughts on that.
Yeah. I think Chetan Puttarajapa from UPMC actually did a cost analysis comparing the cost of biopsies and the cost of cell-free DNA. And I think the commentary even after that is that the cost of biopsies was even underestimated because it didn't include the possibility of complications and things like that. And it's unfortunate because Eurofins actually had a combination biomarker which was combining gene expression with cell-free DNA, OmniGraph. And from what I understand, that needed to be withdrawn largely because of these issues. So I would like to see that thing revisited. Yeah. Thank you for the question. Yeah.
Thank you.
Right. Thank you very much.
Thank you.